I'm Rajesh Uppal, Director IDST. We bring sharp insights and foresight on defense, security, and emerging tech. Let's dive in.
Imagine holding the blueprint of life in your hands. That's what DNA is, a molecule that carries the instructions for everything from your eye color to how your cells fight disease. But today, DNA is more than a blueprint. It's a toolkit, a key that's unlocking new possibilities in medicine, agriculture, forensics, and even national security. Inside every cell, DNA is tightly packed into chromosomes, forming the genome, the complete set of instructions for an organism. DNA is made of repeating units called nucleotides, which include 4 bases: adenine, thymine, guanine, and cytosine. The sequence of these bases acts like a code, directing everything from protein creation to cell behavior. For humans, the genome contains around 3 billion base pairs, forming about 20,000 genes. Each gene is a set of instructions that keeps life running. The revolution began when scientists developed tools to read and manipulate this code. One of the most transformative breakthroughs was PCR, the polymerase chain reaction, a kind of molecular photocopier invented by Kary Mullis. PCR allows scientists to take a tiny fragment of DNA and amplify it millions of times. This technology powered everything from early genetic research to modern COVID-19 testing, letting us detect pathogens, study mutations, and identify individuals from just a few cells. Then came DNA cloning, a way to isolate and copy specific genes. By inserting a piece of DNA into a tiny plasmid and letting bacteria multiply, researchers can produce valuable substances like insulin, growth hormone, and antibodies. The idea is simple but revolutionary. Harness nature's own replication machinery to manufacture medicine. Building on that, recombinant DNA technology took things further, cutting and splicing genetic material from different organisms to create entirely new combinations. Using specialized enzymes like molecular scissors, scientists can insert useful genes into bacteria, plants, or animals. In agriculture, this enabled crops to resist pests, survive herbicides, and yield more food. Bacterial genes can give corn self-defense against pests, reducing pesticide use, or help fruits ripen more slowly, extending shelf life. Recombinant DNA is not just science, it's a bridge between understanding life and shaping it for the greater good. More recently, CRISPR-Cas9 has brought gene editing into the spotlight. This bacterial-inspired system lets scientists insert, delete, or modify genes with astonishing precision. Its applications are vast, curing genetic diseases, engineering disease-resistant crops, and even producing microbes for biofuels. But with this power come ethical challenges, from designer babies to gene editing in humans. The real-world impact of these technologies is already profound. In medicine, recombinant DNA has revolutionized drug production. Human insulin, growth hormones, clotting factors, and cancer therapies are now manufactured using genetically engineered cells. DNA-based vaccines, like those developed for COVID-19, allow the body to generate its own defenses safely and quickly. Gene therapy offers hope for conditions like cystic fibrosis or sickle cell anemia by replacing faulty genes with healthy ones. Microbes, too, have become biofactories. Genetically engineered bacteria can produce medicines, clean up pollutants, and even synthesize cancer-fighting compounds. In agriculture, genetically modified crops are more resilient, reducing reliance on chemicals and helping secure the global food supply. DNA has also transformed forensics and law enforcement. DNA profiling relies on short tandem repeats, unique patterns in our genome, to identify individuals with remarkable accuracy. Today, portable systems allow police to analyze samples at crime scenes within hours. Probabilistic genotyping uses advanced algorithms to interpret complex mixtures, enabling reliable results even from minimal or degraded DNA. Programs like the Belarus-Russia Union States DNA Identification Project are taking this further, exploring the ability to reconstruct a person's appearance and traits from genetic material alone. This fusion of genetics and data science could redefine security and justice, but it also demands serious ethical oversight to protect privacy and prevent misuse. Emerging technologies like DNA microarrays and microfluidic chips are miniaturizing labs, allowing rapid, high-throughput analysis of thousands of genes in minutes. These advances accelerate everything from forensic investigations to personalized medicine, where treatments are tailored to your unique genetic makeup. As we harness these tools, ethical questions loom large. Who should have access to genetic therapies? How do we prevent misuse or discrimination? Where is the line between healing and enhancement? The DNA revolution is no longer a future possibility, it's happening now. From curing diseases and improving crops to solving crimes and understanding human ancestry, the code of life is being read, interpreted, and rewritten. With every discovery, we are not just observing science, we are living it. How we wield this power today will shape the next chapter of life itself. Thanks for watching. Subscribe, follow, or visit idsth.com for more cutting-edge defense and tech insights. See you in the next one.